Biochem Cadet 1.
Biochem Cadet 2.
Sir, fellow cadets, I'm doing my presentation today on glucose 6-phosphatase. So, using technology advantage, present an iPad. So if we're going to go back to what this enzyme actually does, we go back to this wonderful excerpt from our metabolic passages pathway. You can see that the glucose 6-phosphatase is involved in the glucagonate pathway, where it goes from glucose 6-phosphate, taking away the phosphate, bringing it back to glucose, needing this independently in order to allow for the independent regulation of glucogenesis versus glycolysis. What's really unique about this enzyme in particular, and I think we touched lightly on this in class, that this is the one enzyme in these set of pathways that doesn't actually occur in the cytosol. Instead, it's found in the lumen of ER. Or rather, the actual catalytic site is found in the lumen ER. The enzyme itself is actually bound, it's a transmembrane protein bound within the endoplasmic reticulum itself. So what this allows for is separation from all of the above, from everything else, to allow the creation of glucose. And this is important, especially as I discussed in class. Glucose can be readily exchanged in and out of the cell. Glucose 6-phosphate can't. So you need this in a separate regulatory area so that the few instances where you do want this to occur, especially often in the liver, you make sure that you have this separate, so then when you transport it out, it's automatically pretty much going to go out. So, and again, it's the gateway pathway in terms of glucogenesis. Most of the time you have any ready store of glucose stored as glucose 6-phosphate. And then this is the one pathway, and this is the one enzyme that allows it to be taken out and used as glucose. So, moving on to the actual protein itself, what's interesting is that although this is very important, they still haven't been able to actually isolate it from the membrane. They have a topological idea of what actually it looks like, but they don't know what it is specifically. So in terms of primary structure, you can count it here, but it's 357 amino acids long. Secondary structure, the main component you can see are these nine groups that go across the membrane, and these are primarily alpha helices, hydrophobic alpha helices in particular. Obviously, you need a hydrophobic effect. You're in a lipid dual membrane, so all that comes into play. And you also have loops. They're not turns because you don't have the specificity of turns. Instead, they're just 180 degree loops that go back and forth. In terms of tertiary structure is it's mainly, if you look at it, all it is just a repeating helix loop, helix loop, helix loop all the way through. And what's interesting, and then quaternary structure, it's a single polypeptide, so there is actually no quaternary structure. What's interesting, if you look at it, is that, at least in terms of what they identify in this picture, all the black circled amino acids are ones that mutations have been isolated in terms of defective protein, and the few that are shaded in gray and enlarged are ones that are actually involved in the catalytic aspect of the enzyme. Specifically, if they're part of the phosphatase motif that's seen in other similar enzymes. And just to orient you, so to make sure you understand, this is the ER of the lumen, where these two aspects make the primary catalytic region, where they go into a hydrolytic core in between them. And then this is where the primary phosphatase, and you have the hydrolysis of the phosphate off it, and you release glucose and inorganic phosphate. So again, moving on to catalytic activity, they have a few key enzymes that are, rather key amino acids that have been identified as part of the motif. What's really important that they have is cysteine 119, which they believe is involved in the phosphorylation transfer. Then also arginine 96, which is involved in the glycosylation. So these are the ones that are actually allow for the actual transfer and the actual intermittent steps within the catalytic event itself. So what's interesting about this, though, is that regulation they haven't yet found anything that actually activates it or upregulates it. All they found is inhibitors. But if you think back to its purpose, this actually makes practical sense. The reason you're doing this is only if you need glucose and only if you're trying to take away either the glycogen storages or if you're making glucose from, let's say, broken down amino acids or other carbohydrates that are available to you to be exported out to the bloodstream. So really you need to make sure that this doesn't go doesn't occur overactively. So one, we said it's a glucagonic pathway, therefore we would think that insulin naturally would turn it off, and it does. In this case, one of the insulin reactions causes phosphate kinase B alpha in particular, to be activated, which then phosphorylates part of the activator region for the actual gene itself, which then downregulates the gene and then makes less of this enzyme actually created. Another one is the Km value is actually a huge thing. The Km value for this is over an entire order of magnitude. Like the normal G6P available in the cell is 0.05 to 0.1 millimolar, where the Km for this is 2 to 3 millimolar. So it's over an entire order of magnitude, so you really need either glucogenesis or you need glycogen degradation in order to make sufficient quantities of this for this to actually be able to activate to any sufficient extent. They've also found that its products indirectly inhibit it. Also, you think it's going to reverse the direction of the reaction anyways, so that makes practical sense in itself. But in that sense, it's also inhibited by its own products. And then you also have fatty acids and acetyl-CoA, although I'm not entirely sure why. Also inhibit it, and then detergents, but that's more of a man-made aspect. I mean, that's involved more with the degradation of the ER itself, so it's kind of moving beyond the point. Then the current, just leaving it on why is this actually important? It's important because there are a number of diseases that are associated with this. You have to think that this is what's involved, especially with glycogen. The only way, when you go through the glycogen degradation process, you end with G6P. So you need some method in order to make it to glucose, because that's the whole point of what you're doing, is you have that glycogen there for glucose storage. But if this is defective, that doesn't work. So what you end up doing is, they found that glycogen storage diseases, there's, I believe, 11 subsets in total, but they found that many of them are involved actually with this pathway. So for example, type 1A is involved with G6Pase. Type 1B is involved with the G6P transporter. Type 1C is involved with it goes on and on, because there's an entire system, because again, this is an ER of the lumen of the ER, excuse me. So you need glucose to be transported in and out. You need phosphate, you need the inorganic phosphates to be transported in and out. You need all of these to be made into a proper setting so that you can actually have this move forward instead of move backward or just be inhibited because there's too many products in there to actually make to make it useless. So if you take any of the machinery away, you inherently make it so that it doesn't work. So they're looking into this. Also, some aspects potentially, they're thinking that this may be involved in diabetes, because you can't, if it's overactive, then you have too much glucose being pumped out because you're taking so much of the glucose 6-phosphate and then transporting it out because you keep on making it. So they're looking into a lot of things, but this is something that they've actually proven and classified that's specifically designated towards these genes exclusively. So these are my references. So, sir, classmate said in my brief. Are there questions? Yes.
So if you were, say you were in an extended starvation state for a really long time, like on and off, let's say you were like in an impoverished country, would there be problems with this over time? Would it be overworked from the glycogen?
The thing is, you have to think about it, at least in terms of what we talked in class, is you end up taking away all your glycogen storages, you take away all your ready storages. So if anything, like if you go back even to the Michelson's constant, how high it is, you have to make so much G6P, so you have to have that availability. So you're going to cut yourself away from any of that availability over time. So, I mean, at least the way I would suppose, based off the mechanism that this goes, is that it could not be overworked because it requires these substrates in a sufficient quantity, so your body really has to be doing work in order for that to actually occur.
Structural standpoint, do you know how that the protein actually becomes embedded inside the membrane? Like, how is it like manufactured?
For a lot of these, I mean, I think I'd have to go back more to like kind of cell bio type aspect, where usually for those they have the pores going to the ER, the rough ER. You're then making it and you're embedding it through, and then it's just as it's embedding through, it's gonna naturally fold into this conformation. Again, you have the hydrophobic, obviously hydrophilic regions, and then hydrophobic regions. So that's what it's naturally gonna want to be able to do. Anything else? Thank you. That concludes my brief.